Okay, let's get started. So thank you all so much for coming to the very first EF1A2 community conference. We're really delighted with the amount of support and enthusiasm we've had from you all. I'm going to hand over to Heather in a moment, who will do the housekeeping and the first talk. And just before I do that, I just want to say what an enormous amount of work Heather's put into this, and how appreciative we all are of everything she's done, because she's doing it, all this tireless amount of work, alongside doing her PhD with all the lab work. So I will hand over to Heather now and see you again in 10 minutes.
Thanks, Kathy. I'm just going to share my screen. Okay, so yeah, thanks very much for coming along. These are just a few notes for attendees, maybe if you haven't used Zoom webinars before. So first I want to point out the chat and the Q&A functions. So you should be able to access those at the bottom of your screen using these icons. You can use the Q&A to post questions during the talks that you want the speakers to answer later. And feel free to use the chat to post other things. So maybe, where you're listening from, for example, but please don't use the chat to post questions for speakers, because then we might miss your question if we're looking at the Q&A. In the interest of keeping things kind of streamlined and running to time, we're doing a Q&A session at 6 p.m. where all of the speakers will go through some of the submitted questions and answer them. But unfortunately, speakers won't be able to answer specific medical questions or give advice about individual cases, but we're happy to try and answer any other questions that you might have. So we do have a very mixed audience here. So for some people, some of the information that you get tonight might be really familiar already, but some of it might be brand new. But mostly we just hope that there's something for everyone here. We have breaks scheduled at 4:45 and 5:45, and we'll do our best to stick to that schedule, just to make sure that people can step away from their screen, get a drink or anything else, and just refocus. And we're also recording these talks, and they'll be shared online afterwards. So if you need to step away, or if you miss something, don't worry, you'll be able to catch up later. So finally, at the end of the session, I'm going to share a feedback form link with a QR code on the screen, and later on by email. And it'd be really good if you can fill that out so that we can see what went well and where we might need to improve things for future events. Okay, so my talk is going to be a kind of primer on EEF1A2 related neurodevelopmental disorder. It's partly to bring everyone up to speed because, as I said, we have a mixed audience. So some people will be quite familiar already. And it's also partly to give some background on the science behind it all. So the official name is EEF1A2 related neurodevelopmental disorder, but that's quite wordy, so I'm going to call it EEF1A2 syndrome from now on, just for the sake of being brief, and because that's a name that some people are already familiar with. So EEF1A2 syndrome is a condition that typically causes epilepsy or seizures, developmental delay, and intellectual disability. And one key feature of this condition is that it's very variable. So it can range from being quite mild to very severe. So, for example, most children who are affected, but not all, will start to have seizures within the first 4 months after birth, but this can start later on as well. Some people will respond really well to treatment with standard anti-seizure medications, but in other cases, the seizures can be more difficult to control. There can be problems with movement that range from low muscle tone, coordination issues, all the way to being unable to walk or to sit up. And some people who are affected might be non-verbal, while others communicate in sentences or with signs. So each person who's affected might have a very different experience of the condition. So it's a very rare condition. We know of about 200 cases worldwide, and we hear about these both through published scientific papers and case studies, and then we also hear about diagnoses through personal communication. So, for example, we hear about things through the support group or by emails to the lab. But there was one paper that was published fairly recently that estimated that this condition occurs just under 3 times per every 100,000 births. So although that's still a very small number, it's more than the cases that we already know about might suggest. We've only known about EEF1A2 syndrome since 2012, so this is still a relatively new condition in that sense, and many of the people that we know of who are affected are children. But we suspect that there are probably a lot of adults who might have been diagnosed with epilepsy or intellectual disability well before 2012, or have a kind of milder presentation, who never had a genetic test and so were never diagnosed with EEF1E2 syndrome. And although there's been a lot of progress with people receiving genetic diagnoses over the last few years, that's mostly been in paediatric settings in children. But we are hearing about new cases all the time as genetic testing becomes more affordable and accessible. So this condition has quite a mouthful of a name, so I thought I would tell you about what EEF1E2 actually is. But first, I just want to go over some biology terms so that everyone is on the same page. Sorry, I've got a bit of a cough. Sorry. So firstly, the cells are the fundamental unit of human life. There are over 200 different types in the human body, and they make up all of our different organs and tissues. They perform different functions, and those cells can look very different from each other. Cells are made up of proteins. Proteins can act alone or bind to one, two, or many others to form larger structures. Proteins themselves are made up of amino acids. They're the base components out of which all proteins are constructed. And DNA is like the cell's instruction book for making new proteins. It contains the code for every protein that's required by every type of cell, so which amino acids the protein is made of and what order they need to be arranged in. Part of what allows cells to have all these different structures and functions is by having the code for different proteins turned on or off. And when we talk about genes, we're referring to just sections of DNA that code for different things. So by following the instructions encoded in DNA, cells can create any new protein that they need. And the process of making new proteins in a cell is called translation. Sorry about that. So the amino acids are assembled into the correct order, following the instructions encoded in DNA, and then they're folded up into a three-dimensional structure. And all cells are completely dependent on the process of translation for survival and for normal function. Sorry, my slides are just not changing. There we go. So now I can tell you that the gene EEF1E2 codes for the protein EEF1E2 with a small e, which is why you might see these written differently if you're reading about it online. The protein EEF1E2 isn't made in every cell in the body, but only in a specific few types. It's found in the neurons, which are nerve cells that send signals all over the body that control everything from breathing, talking, walking, and thinking. It's also found in the skeletal muscle cells, which are what you think of when you think about muscles, your biceps, your quads and abs, and the slightly different cardiac muscle that makes up your heart. But what does EEF1E2 actually do? Well, it's a translation factor, and that means that it plays a really important role in that process of translation of making new proteins. And the interesting thing is that a very similar but slightly different protein called EEF1E1 does this job in every other cell type in the body. And that suggests there's something special about EEF1E2 that we need in the nerve and muscle cells specifically, but we still don't fully understand what that is. But what we do know is that EEF1E2 is vital for survival and for normal function. So EEF1E2 syndrome itself is caused by harmful changes in the DNA that codes for the protein EEF1E2. And each of the mutations that cause this condition change just a single one of the amino acids out of the nearly 500 that make up the EEF1E2 protein. In almost every case, these mutations are de novo, and that means that they weren't passed down from an affected parent, but they've happened spontaneously in an egg or a sperm cell. And that means that the chances of future children of the same parents having the same condition is really low. So you may have seen this kind of notation before in relation to an EEF1E2 diagnosis. And this is just a way of describing a genetic mutation by explaining what has changed either in the DNA or the protein. So the numbers represent the position of the change and the letters tell us what has been changed. So in this case, a change in the DNA code on the first line means that the correct amino acid, glycine, which is number 70 of the amino acids in the sequence, has instead been replaced by a different one called serine, which is where we get G70S. This means that the protein then gets made containing a mistake that might affect how it functions. So these two codes represent the same end result, but they just describe it at different levels, one at the DNA level and one at the protein level. So we do have a clear understanding of what's being changed in the DNA and the protein, but what we're still trying to understand is how exactly the different genetic mutations actually affect the way that EEF1A2 works. So I mentioned earlier that it's a very variable condition with a really wide range in severity, and we think that that's partly because there are many different mutations that have been found in people with EEF1A2 syndrome. There's over 50 different mutations in only around 200 people, and some are more common than others, which means that quite a few of these mutations are only occurring in one person in the whole world. And that means that it's very difficult to predict how someone will be affected without understanding exactly what that mutation is doing to EEF1A2. So you might have seen this analogy before if you've looked at Kathy's website at all, but we each have 2 copies of the gene that makes EEF1A2 that we need to keep us healthy. But it can help if instead of thinking about genes, we imagine that we need 2 doses of EEF1A2 every day to keep us healthy and stop us from getting seizures or epilepsy. So if you picture 2 pills that contain the correct amount of EEF1A2, one is not enough. We need both doses a day. So now if you imagine what happens if you have a mutation in one copy of EEF1A2, you still get 1 dose per day that works well, which is the normal copy of the gene that produces the right amount of functional EEF1A2, but then the other capsule isn't quite right. So some people might have a mutation that means although they're still getting 1 normal dose of EEF1A2, the second dose isn't enough, and that's what we would call a loss of function mutation. So that means that they don't have quite enough EEF1A2 to keep them healthy and prevent epilepsy. And in this case, their symptoms are only caused by the lack of EEF1A2, by not having enough. Other people might have a mutation which means that they have the right amount of EEF1A2 in their second dose, but it's not quite the right drug. So in this case, not only is the person not getting enough EEF1A2 from their single healthy dose, but the mutations mean that the EEF1A2 in their second dose could be toxic and actively harmful to the nerve and muscle cells. And we call this a gain of function mutation. And in this case, the person can have a kind of additive stacking effect of both having a lower dose than they need to stay healthy, combined with the harm caused by the mutated EEF1A2. So there are lots of different variations of this scenario, different combinations of loss and gain of function, and different ways that EEF1A2 might be affected. So, for example, EEF1A2 needs to be able to interact with other proteins to do its job. Mutations can affect protein interactions in more than one way. So it could sit on an area of EEF1A2 where it interacts with another protein and it either partially or completely stops this from happening. And then that could stop EEF1A2 from being able to carry out its normal functions. It's even possible that a mutation could allow EEF1A2 to bind to a new protein that it previously couldn't interact with, and that might allow new functions which could potentially be harmful. Oh, my slides are jammed. And mutations in EEF1A2 could prevent it from correctly folding into its final protein structure after all the amino acids have been assembled, maybe making it unstable or making it clump together, which can be damaging. So there are multiple different ways that EEF1A2 can be affected by mutations. And then these effects of EEF1A2 inside the cells then have wider impacts on bigger systems like the brain, nervous system, and muscle function. And that leads to the symptoms that we see, like seizures. So a lot of research is devoted to understanding exactly how mutations translate into symptoms, because we hope that there's some way of correcting that or to compensate for it and to improve people's symptoms. And so the ultimate goal of this kind of research is to find treatments or even potentially a cure for EEF1A2 syndrome down the line. But at the moment, there is no cure for EEF1E2 syndrome, and instead the symptoms are typically treated or managed individually. So for example, epilepsy can sometimes be managed using anti-seizure medication, and other therapies such as physical or speech therapy can be used to improve movement and communication. Sometimes medications for other symptoms are prescribed, like sleep or gastrointestinal problems. And people who are affected can be supported with the use of things like communication or mobility aids as well. But overall, the response to treatment is also very varied, and what works well for one person might not suit another person. So a really individual approach is usually needed for finding the best treatments for someone with EEF1E2 syndrome. With rare conditions like this, lots of people call the search for answers a diagnostic odyssey, because it can be quite difficult to get a diagnosis. But an EEF1E2 diagnosis is confirmed by genetic testing, usually after symptoms have been noticed and there hasn't been another cause found for them. Testing is really highly recommended for people with epilepsy anyway to determine the root cause, because it can have big implications for how treatment happens. And this is most often done by taking a blood sample, but in some cases it can be done with a saliva or cheek swab. And the test is likely to be part of a panel test. It won't just be looking for EEF1E2 usually, it'll be looking for all different kinds of genetic epilepsy. But ideally, any child who has seizures or developmental delay should be considered for testing for EEF1E2, even if they are like well-controlled seizures or it's a milder presentation, because it may be the cause. So as a final note, I just want to talk about the relationship between EEF1E2 and cancer. So if you've ever searched online for EEF1E2, you might have noticed that lots of different search results about cancer will pop up, but that is not related to EEF1E2 syndrome. So EEF1E2 is supposed to be produced in nerve and muscle cells, but in some cancers, it's wrongly produced in cells that it doesn't belong in. And then that contributes to cancerous cell growth because it's making extra protein, essentially. And that's not connected to EEF1E2 syndrome or the related DNA changes. So just to summarize, EEF1E2 related neurodevelopmental disorder is a really rare genetic condition that's caused by harmful changes in the DNA that codes for the protein EEF1E2. And EEF1E2 plays a really important role in making new proteins in the cell. The condition can really vary. It can be really mild or very severe, but the typical presentation that we see is epilepsy, intellectual disability, and developmental delay to different extents. It's diagnosed by genetic testing, and although there's currently no cure, symptoms are often treated individually, but they do require a tailored approach. Thank you very much. I just want to quickly acknowledge my lab group, my supervisors and my research funders. If you have any questions, just ask them in the Q&A and I would be happy to answer them. And we'll be hearing from a few of the people on this list of lab members about what they do in the lab and the kinds of research questions that they're trying to answer. But first, Kathy will be given a talk on research challenges and opportunities for EEF1E2. And for those of you who don't already know Kathy, Professor Kathy Abbott is the head of a lab at the University of Edinburgh that investigates EEF1E2 and EEF1E2 syndrome.
Right, thank you very much for letting me do this talk. So one of the things that came up in the survey that Heather did, asking people about what they wanted to hear about, was how we do research. So I'm going to just spend the next 10 minutes just talking about very high level sort of stuff about the way we do research in the lab and the particular challenges and opportunities for EF1A2. My slides aren't changing. Right. So I was going to just talk about the funding landscape very briefly. So as you may know, an awful lot of the medical research, at least the preclinical medical research that goes on worldwide, goes on in university settings. But we don't have direct funding for this in labs. Scientists have to apply for grants. And you can apply to various places. So there is government funding. In the UK, this is mainly through the Medical Research Council, and the US has the NIH, and there are equivalents in other countries. But it's quite a burdensome process applying for these grants. It's very expensive because you have to cost in all the expenses that the university incurs. And importantly, there's a very low success rate. So it's not a trivial thing to keep labs going. There are smaller charities. Cure Epilepsy is a relevant one in the US. We have support from Action Medical Research in the UK and Epilepsy Research Institute. And they tend to give smaller amounts of money, but very much more focused. So it's much easier to approach them for funding. And then the research in labs is generally carried out by a whole range of people at different career stages. So you have postdocs who already have PhDs, who are often supervising a lot of the work. PhD students like Heather, and you'll hear from Tamsin as well in a moment. Technicians who are obviously keep everything going. And then a surprising amount is actually done by project students who may only be in the lab for 10 weeks, 6 months, up to a year. And many of them contribute enormously to the work that goes on. So we use a range of different approaches to doing work. We mainly use molecular biology, which means just really looking at proteins directly. And this is a sort of cartoon of the way we look at proteins by staining them with antibodies. We do proteomics, which means looking at all of the proteins that there are in a cell and the way they're changing when you have a mutation in EF1A2, and that's supplemented by biochemistry. We do computational modelling so that we can try and look at the effects and predict the effects of mutations on the protein structure. We grow cells in culture. That means you just have a Petri dish and you can the cells will just sit and divide. We do a lot of microscopy, and you can see the picture there is neurons in the brain, and the red staining is EF1A2 in the neurons. And this picture was made by Faith Davis, who's in the audience. And then we also make genetically modified mouse models. And this bit is really, really important for finding out about exactly how the mutations work, but very importantly for any kind of drug testing or treatment testing subsequently. So there are a number of challenges, particularly with EF1A2, and this is not making excuses, but just to set the scene. So one issue, as you all are well aware, is the very variable presentation. So not everybody has seizures. Some people can talk, some people can't. Some people can walk, some people can't. There's evidence for a gain of function, which is what Heather was talking about in her talk, where the gene works, but it makes a protein that causes damage to the neurons when it's there. And that requires a completely different approach when you're thinking about therapy. We can't reproduce any of these symptoms in cells and culture, so we can use them for looking at sort of biochemical molecular things, but you know, the cells themselves don't reproduce any of the obviously seizures or behavioral changes. And even when we've made the mouse model, seizures are not easy to study because, of course, they happen sporadically. And when you just go and look at a mouse in a cage, most of the time it won't be having a seizure. So it might be having a seizure at night when you're not there. And that has given us another whole sort of range of challenges that can be addressed and are being addressed. The biggest challenge probably, as you've heard, is just the number of different variants there are. And this is a word cloud that sort of reflects the frequency of the different changes that we see at the protein level. And one of the issues is, will we need models for each variant? We hope not. We're trying to find ways of grouping mutations together so that we can address things in a much more rational way. But obviously, as Heather mentioned, you know, huge numbers of your children will be the only one with that particular mutation. So there are various types of potential therapy. As you all know, anti-seizure medication is widely available. It's in use. And that can be incredibly useful. It doesn't actually address the underlying problems. We might be able to treat the underlying disorder by changing the amount of EF1A2 in cells. And then there's a sort of genetic correction approach. So the three main approaches would be gene therapy, where you would swamp the nerve cells with normal EF1A2. And that's an incredibly powerful approach for many, many disorders. And you'll be hearing more about Rett syndrome later from Monica. And that's exactly what you need there, because that's a loss of function disorder. Drug repurposing, you're going to hear a bit more about from Tamsin in a minute, on screening small molecule libraries. These are drugs that have known safety profiles, and they're relatively cheap, but we don't know whether upregulation of EF1A2 would actually improve or even make the condition worse. So this needs a lot more work. And then finally, one of the other approaches that we're working on now is something called allele-specific antisense oligonucleotides, which doesn't trip off the tongue, but the idea is this would stop the mutant protein from being made. The problem is you have to redesign and test for every variant, but it's an approach that's showing a lot of promise in other disorders. Having said all the challenges, I just want to talk about the opportunities. So we've got a lot going for us in this field. So in our lab, we've got expertise going back 25 years. This was the first paper I published on the EF1A2 in the same month my daughter was born, and she's now nearly 26. We have a lot of collaborations worldwide, a lot of people very interested in the EF1A2. We've got these bespoke mouse models. You'll hear about one of them from Grant in a moment. There are more and more people researching EF1A2 worldwide, and this is, you know, this is a hugely positive step. It's attracting a lot more interest than it was 10 years ago. There's enormously rapid progress in genetic technology. We can do things now in the lab that you couldn't have dreamt of 10 years ago, and it's just improving all the time. It's really exciting times to be in. And then, as you'll hear from some of the other talks, and you've already heard from Heather, we've got a lot of inspiring, committed, very bright young people who are devoting their time to researching EF1A2. EF1A2 is not alone, of course. There are a lot of rare epilepsies, and there's been a move in recent years to try and group some of these rare epilepsies together and come together for sort of advocacy and fundraising. There's a Rare Epilepsy Network in the US, for example, and UK RET, which is rare epilepsies together. And finally, as I said, there are all these incredible people who are imaginative and brilliant and doing fantastic work. Not all of them are still in the lab, but these are the people who've contributed a lot directly to EF1A2 related neurodevelopmental disorder. And then finally, I just want to thank all of you, because, you know, that's why we get out of bed in the morning.
My name's Grant. I'm a postdoctoral researcher in Kathy's group at the University of Edinburgh. And today I'll be sharing some data that I collected during my PhD on a mouse model of EF1A2 syndrome, which I developed. So just briefly, how can mice help with research into EF1A2? So our main overarching aims, or one of them, is to understand the pathological mechanisms of these mutations so that we can design appropriate therapies. So one of the big questions is whether they operate through loss of function, gain of function, or both. And we also want to develop disease models so that we can test those therapies. And mice can help us with both of these. So just like humans, mice have an EF1A2 gene, which is used in the same cells as in humans. Mice can also have many of the same seizure types that are seen in humans, and they also have correlates of human neurodevelopmental disorders. So for example, you can have mice with difficulties in learning and memory. And we can also compare the phenotypes of mice carrying the missense mutations to mice which have no mutation, no EF1A2 at all, to sort of see whether the missense mutations behave like mutations where there's no EF1A2, which lets us see whether they operate as loss of function. So I had to first choose a mutation to model in mice, and I selected mutation E122K as it was the most commonly identified at the time and still is, and it also has a consistently severe presentation in patients. There are 11 published cases of the E122K mutation at the minute, and these patients show motor abnormalities, so delayed psychomotor development. 4 are non-ambulatory, which means that they don't walk, and 6 patients show hypotonia or low muscle tone. All the patients have epilepsy, with a typical onset in the first years of life, but often the first months, and they show a diverse range of seizure types with variable seizure control. All these patients also have moderate to severe intellectual disability. They're typically non-verbal, and autism is also common. So I used a tool called CRISPR-Cas9 to introduce the E122K mutation into mice. And this here is a Western blot showing the EF182 protein. And in the first 3 lanes, or the first 3 green bands, you can see the wild-type protein. Then we have the protein where the 1 copy of the gene has been changed to E122K. And in the next 3 lanes where both copies of the gene have been changed to E122K. And that's just to show you that the E122K mutation doesn't cause the protein to disappear or change in size. It's still detectable in the brain. The first main question was whether mice carrying the E122K mutation show motor abnormalities, such as are seen in patients. So I used a simple scoring system called the NeuroScore to do this. So in this, we simply look at the animal's gait or the quality of its movement. We check hind limb muscle tone. We score their ability to walk along the ledge, and we check for abnormal curvature of the spine. And this gives us the NeuroScore. And what's important to know about this score is that a higher score is worse. So here in this graph, I've plotted the NeuroScore of wild-type mice from 14 days of age through to 28 days of age. And you can see they start with a moderate NeuroScore of around 3 or 4, which quickly drops to 0 as the mice develop and learn the various tasks of the NeuroScore. Next, here in blue, I've got mice which have no EEF1A2 at all. Now, you remember that Heather mentioned that EF1A2 is essential for life. And what you can see is that these mice start off with a score that's similar to wild type, but around 3 weeks of age, the score goes up and up and up as the mice begin to deteriorate due to missing EF1A2 in neurons. Lastly, here in pink, I have mice which have only E122K EEF1A2, and you'll see that they start off with a higher score than all the other mice, but then they go on to outlive the nulls, and they die by about 4 weeks of age. So what does this picture tell us? Well, we can see that mice with E122K mutation have worse motor function than wild type. Their score is higher, which means that they do have motor abnormalities similar to those seen in patients. We also see that mice with E122K EF1A2 live for longer than mice with no EEF1A2 at all. And this tells us that E122K does not cause complete loss of function. And then lastly, we can see that mice are better off having no EEF1A2 at all than they are having E122K. So this pink line is higher than the blue line at first. And this tells us that E122K is toxic in some way, causing a gain of function. The next big question we had was whether mice carrying E122K have seizures that are such as are seen in patients. It's first of all important to say that we didn't notice any seizures when we were just studying the mice or observing the mice. But as Kathy mentioned, seizures in mice can be very difficult to spot. For example, they can be very brief, or they can happen when the mice are not under direct observation. In humans, we use electroencephalography, or EEG, as the gold standard technique to detect and quantify seizures. So we sort of look at brain waves, if you like. And we can actually, you may be surprised to learn, do the same thing in mice with some fiddly surgeries. So what we did is we took a bunch of adult wild-type mice and a bunch of mice which were heterozygous for E122K, like patients, meaning that 1 copy was E122K and 1 copy was normal, and we performed EEG recordings on these mice. So these are the sort of brain waves that we were able to capture from mice. So you can see that they have different waveforms when the mice are awake versus when mice are in non-REM sleep or REM sleep. As we found previously, the mice were not having obvious seizures, for example, freezing or experiencing convulsions during these recordings, which wasn't a big surprise. The EEG was also mostly normal. However, we did notice some abnormalities. So the main abnormality that we noticed were these high amplitude spike trains, which occurred in the cortex of the mice. And you can see that here in this trace. These events occurred in all of the mutants tested, and they only occurred in the mutant mice, so they're definitely caused by the mutation. They occurred when mice were awake as well as when they were asleep, and they occurred on average 6 times per hour, and they lasted for just under 1 second on average. However, the mice showed no outward behavioral signs during these events when they occurred. during wakefulness. And so what they think these are is electrographic seizures, which is to say seizures which can only be detected by EEG. So just to sum up, the mice have been able to help us understand the pathological mechanisms of E122K. We saw that mice were better off having no EF182 at all than having E122K, which tells us that there's a toxic gain of function, and that's really important for designing therapies for E122K. And we also want to develop disease models to test therapies. Now, since these mice have relevant motor abnormalities and electrographic seizures, that tells us that they're capturing some of the pathology that's seen in patients with E122K, and that we can use these mice to test therapies in the future. All of this information and much more is available in a paper that I recently published this year. So do check it out if you want to know more about the mouse model of E122K. And then thanks very much for listening. I'd also like to thank Kathy and all the members of the Abbott group, past and present. Also, I'd like to thank Alfredo, who taught me the EEG surgery, and thank you to our funders as well.
Hello, everyone. I'm Cavan. I'm just going to try and give you an idea of how we try and work in the lab to look at the EF182 mutations. So we use a range of techniques to try and look at EF182. So my work is focusing on how do the mutations affect EF182 function. And we use a range of different lab techniques. These involve looking at cells and the mice models that Grant has just talked about. But we also use computer programs to try and model the mutations. So how might this be useful? So we know that certain parts of the EF182 protein are particularly important for its function. And so knowing where a mutation is and what it's nearby can be a really useful way to predict what effect the mutations might have. So we show you the EF182 protein as the amino acid sequence here, but this is a bit of an oversimplification. As in cells, this string of amino acids is folded into a three-dimensional structure, like so. So if we know the structure, we can then model it using computational programs, and we would show the folded structure of EF182 like so. And here I've just shown a little mutation, a mutated amino acid in red here, so you can see what it looks like. And what this means is that two amino acids that might not appear to be close together on the sequence, the amino acid sequence might actually be very close together. And so I'll give you an example. So here we have a mutated amino acid, so a mutant in red on the EF182 protein. And in yellow, we have three amino acids that form a binding site. So they're a very important area for the EF182 function. And we might not expect from the sequence that they're very close together, but when we fold the protein, we show that they're actually right next to each other. And so we might expect this mutation to be very disruptive for EF182. And as you know, there are many different EF182 mutations. And so by modeling where each one of them is, it can be a really useful way to predict what effect the mutation might have. So we can see which mutations are nearby to each other and which mutations are nearby to important parts of the protein. And this can be useful for guiding what lab experiments we carry out. And hopefully in the future, it will be able to guide therapies and say which mutation, so which treatment might be most likely to be effective for each individual mutation. So just to recap, the function of EF182, so as we've heard, the main job for this protein, the main job for EF182 is to deliver the building blocks that make up new proteins. So without EF182, cells aren't able to make new proteins. And these proteins are what makes things happen in the cell. So they carry out almost every major function. So it's a very important job. And what we don't know is how do the mutant forms of EEF182, how well are they able to make new proteins? Are they affected? And if they are affected, is this the same for each individual mutation? So one way we can look at this is through a technique called mass spectrometry. So if we imagine that EF182 isn't doing its job properly, and its job is to make new proteins, we'd expect that maybe in the brain, we wouldn't see as many proteins, or the proteins that we do see might be different. So what we can do is we can take the brain of a mouse model, for example, the E122K mouse model that Grant has just talked about, and we can run it through this technique, and we can detect and count all the proteins in the brain, and then we can see what has changed. And so you don't have to understand this plot, but each single dot is a protein in the brain. And so the ones that are really high up or far out to the side have changed more. And it's interesting to see, so this can give us a lot of information about how the mutation is affecting pathways in the brain. And you might recognize some of the proteins that have changed here that I've labeled, as they're often, some are involved in other developmental disorders. And that's because we do see a lot of overlap. So this can be really useful. And if we run this with different mutations, so brain samples from different mutations, and we see certain proteins are changing in both, it can be really useful for giving us targets for possible drug treatments and also markers so we can test these drugs in mice. So that's just an example of some of the ways we look at EF22. Thanks very much. I hope you found this useful. Thanks to the organisers, and I'm happy to answer questions in the Q&A. Enjoy the rest of the conference.
Hi everyone, I'm Tamsin. I'm another student in Kathy's lab, and today I'm going to talk a little bit about drug repurposing, sort of in context of EF102, because that's really where my PhD project comes in. I've been here about 1.5 years so far. So I think when a lot of us think about drug development, we think about it in quite a linear sense. So we're probably going to start off by thinking, I've got my disease, I'm going to find out what causes my disease, and then I'm going to find a new drug that specifically targets that. However, we all know that the body is really, really complicated, lots of different pathways, and we're all aware that most drugs have lots of side effects. And that's because a drug isn't going to do one thing in the body, it's going to be interacting in lots of different ways. And sometimes we can exploit those side effect interactions to kind of be the primary reason we can use a drug for another condition. That's sort of called drug repurposing. So sometimes this is very straightforward. Got lots of examples here of all of the drugs I'm showing at the moment are either in late stage clinical trials or are currently being used as therapies for different rare diseases. So sometimes we're going right from, right there, I've got a drug that was originally used to treat cancer, and then it's identified as a possible treatment for Hutchinson-Gilford progeria syndrome, and that's being used at the moment. And I'll talk a little bit later about how we find these drugs, but sometimes this process is a little bit more complex. So it just appeared there. I've got a drug that was originally repurposed to be used in cystic fibrosis, and then is actually later on repurposed again as a pneumonia treatment. And then sometimes you've got to put a lot more information to get these repurposed drugs. So the drug I've just shown there in the middle was originally a possible therapy for Angelman syndrome. That was unfortunately not a successful endeavor, but was later on found that this drug can be effective at treating Fragile X syndrome when you combine it with a therapy that was repurposed from being an insomnia drug. And then finally, sometimes we might repurpose drugs that aren't actually in the clinic. So the drug I've just shown there, everolimus, was not actually ever prescribed for anything, but will have been held in probably like a pharmaceutical company's database, and then is found later on to be useful for tuberous sclerosis and some forms of brain cancer. So I'm going to talk about why we would drug repurpose. So we've got lots of examples where it's worked, but why would we choose to repurpose drugs rather than make new ones? And the big thing is the time and the money that it takes. So what I've shown there on the screen is what a typical drug discovery network pathway looks like. It'll take a very, very long time. I'm going to start off with over 20,000 compounds, and at the end of it, we might get one drug. But most compounds that researchers and drug companies look at to start off with will never, ever end up in the clinic. So drug repurposing can cut out a lot of that time. So we're coming in right there about halfway through at that screening and validation stage. So you've cut out a lot of that early research, removed a lot of that time. And that's right where my project comes in, is at that screening and validation stage. That's what I'm looking at the moment. And it can really cut down the amount of time it takes for us to find a drug. So basic drug discoveries can take about 10 up to 20 years. With drug repurposing, can still take a long time because there will be challenges, but you can cut that down for about 3 to 12. And if we're taking less time to discover drugs, we're pretty much always going to be using less money, which is a really promising concept for rare diseases. There's lots of different ways that we can find drugs to repurpose. So where my project is really focused is this known molecular target that I've got there on the right-hand side, because I'm specifically always going to be looking at EF102. And I'm doing that using cell screening models. And I've actually got a picture there of some cells that I've made that have the EF102 protein in them, with lots of a few other proteins that give them different colors that make them quite good for sort of high throughput screening of lots of different drugs. But you can use other approaches, and other people in the lab do, and other rare disease researchers. So you can do this known target screening virtually, and that would be where you might have a computer that's going to predict which drugs are going to match onto certain parts of a protein. And you can use mouse models like Grant talked about. You might not be looking at a particular molecular target, but instead you might want to know about a symptom, like does a drug reduce seizures or does it improve sleep? You could do that without actually understanding that molecular mechanism. Then we've got these big data sets that exist for researchers to use. So you've got data about the proteins in different people and biological pathways, and then big databases of drugs. And most researchers that work on drug repurposing in rare diseases are going to combine lots of these different techniques all together to find a drug at the end that would go on to be a potential therapy. So like with anything, there's always going to be challenges that come specifically with drug repurposing, as well as its own unique advantages. So a big one is less risk of failure at that clinical trial stage. And that's going to be because they might have already gone through safety profiling. Manufacturing and supply chains are already available, especially if it's a drug that's currently being prescribed a lot. You're already going to have those supply chains in place. Clinical trials will always have better engagement with drug repurposing. That's been shown. It's because of those safety profiles. And then that big one is faster development time. In terms of a few of those unique challenges that you might get, so you might get some gaps in preclinical data. And that would be where you maybe identify a drug that you think would be a possible option to take through to a clinical trial. But when you apply for that, the regulators say, actually, I want you to go back and put it in some more different mouse models or try some more cell assays to fill in some of that gap before you start a trial. Dose and formulation. So you might be looking at drugs that are currently only used in adults. And if you want to use that for a treatment in children, you're going to need to change that dosage. Then formulation might be, you've got a drug that we take orally at the moment, but actually for a certain condition, it might be better off through the bloodstream. And all of those options are going to need to be tested and checked properly. Then a few more bureaucratic things that can come up with repurposing drugs is unlicensed and off-label access and intellectual property can sometimes come into play if those drugs are currently still made by only certain companies. So just to say thank you all so much for listening. I'll be sticking around for the Q&A if you've got any questions, and I hope you enjoyed the talk.
Hi everyone, that's us back for the next session. So we'll be hearing from Professor Richard Chin. Richard is Professor of Pediatric Neurology and Clinical Epidemiology at the University of Edinburgh, and that means that he studies the presentation, occurrence and management of childhood epilepsy. So Richard, you can go ahead and I'll come off.
Thanks a lot, Heather, and thank you for inviting me to speak, and it's a great opportunity, really. I must first also apologize for any squeaky sounds that you might hear in the background. That might be the family cat who is trying to intervene and participate in the presentation as well. I'll just share my screen now. Hopefully that comes up okay. Hopefully that's okay. So again, because of the survey that Heather sent out, I'm going to be concentrating on the clinical aspect of things and responding to the questions that have been set from the survey. And it's largely related to seizures. What are seizures? What are the causes of seizures? What are the treatments? But I also wanted to include some of the forward-thinking and forward-looking things that are being developed that can help with clinical management. So these are some of the questions that are often asked in clinical practice, and that is borne out by the survey. What is epilepsy? What is a seizure? And there are different ways that you could say what is a seizure, which I'll come to. What types of seizures? What are they called? What are their names? And what are different types of epilepsy are there? How do you confirm the diagnosis of epilepsy? And what is the cause? In this situation, we know that we're specifically about EEF1A2, but we know that genetic causes are increasingly being recognized as causes for epilepsy. And so if there is one test that I would suggest, aside from the MRI, would be genetic testing. Obviously, best treatment is a very hot topic, and Kathy has touched a little bit on that regarding gene therapy. and the whole debate about anti-seizure medication rather than anti-condition treatment. So do we actually treat symptoms, i.e. the seizures, or do we actually treat the underlying condition itself? And what does the future hold? So what is epilepsy? So normally when we think of epilepsy, we think of 2 unprovoked seizures occurring at least 24 hours apart. So if we have multiple seizures all occurring in the same day, then we would still only count that as 1. We see a pattern evolving when you have repeated seizures, as that predisposition to having seizures is what really is the diagnostic criteria for epilepsy. So even if you have 1 single unprovoked seizure and you have a high risk of having another seizure, then we would say that you have an epilepsy diagnosis. And if you have a specific epilepsy syndrome, there are more than 40 types in which we can look at the way that the seizure looks, look at the clinical characteristics, and look at the EEG. We can start to put together a sort of story. So for example, we had someone who had early onset epilepsy, myoclonic seizures within the first 4 months. And we did genetic testing and we saw EEF1A2, then we would say that person is most likely would have a EEF1A2 syndrome. And it is that excessive and abnormal brain electrical activity which causes the seizures. And we can say that the seizures can be described as focal, meaning that it starts on one side of the brain and tends to remain concentrated on one side of the brain, or it can be generalized, occurring across the brain at all at the same time. And the features will depend on the affected brain region. You might hear from my accent that I'm Jamaican, and you can see an example here of Usain Bolt in his typical pose. But sometimes you can see that pose is a characteristic pose that you can get in a frontal lobe epilepsy as well. If it is seizures arising from the back of the brain, that's the area which is responsible for visual interpretation. And so if you have seizures arising from the brain at the back, towards the back, then the seizure type manifestation may be primarily that of visual symptoms. And it's important to note here that, not everything that moves is a seizure. And these are just some examples here of 3 different types of children. And they all have movement abnormalities here. And actually, only one of them is actually a true epileptic seizure. So not every abnormal movements can be a seizure. And so it's very important to know the story, know the history behind it, and actually having a video of it, it is extremely helpful. So video examples of seizure types. I thought I would show 4 different types. Myoclonic seizures. I'm aware that there would be people in this conference whose children who already have epilepsy, and there are others who their children don't have any seizures at all. And so getting an indication of what those might look like would be very helpful. Most of the onset of seizures tend to be myoclonic in nature, and this is a website, epilepsydiagnosis.org. It's courtesy of the International League Against Epilepsy, which is a leading organization against epilepsy worldwide. So if I click on each one of these, it will take us to a video link on this website, and you can also go onto that website and access it yourself. You need a registration, which is free, but you can see all the different types of seizures there as well. So I'll show that again. It just is at 3 seconds, and it's a sudden jerk that you see, primarily in the trunk and in the hands. Okay, and so that was the myoclonic seizure. Let's see if I can find the other one. is an atonic seizure. So you can see the sudden head flexion and head nod that you'll see there. Right towards the end, one more time. Okay, so that's an atonic seizure or a head drop. And this one is a combination of a clonic and tonic-clonic seizure. So you'll see different phases of it. A clonic seizure will be the shaking episodes, and then the tonic phase. The tonic phase of that will be where the person looks as if they're very tense. And then the clonic period is when you start to see the shaking again. So initial start of a clonic phase, then start to emerge in a tonic phase, almost as if they're locked into position. And then we start seeing the clonic phase again, which is high frequency, low amplitude. Right, and I think I'll stop there, and I'm sorry if that it was very distressing, but it was requested, and you know, looking at this website, you can see a number of different types as well. So suffice it to say that it's very difficult to sometimes to know exactly what's a seizure, especially if you have a movement disorder. So if there's one thing that I would say as well that helps us a lot is to take a video. So taking a video is remarkably helpful. And we have the system here in the UK, which is called vCreate Neuro. In this system, what we have is a cloud-based video system in which parents can upload or carers can upload the videos onto the cloud. Then the physicians can access that. It becomes a link through their medical records that we can access it, and we can see all the videos in real time, and then we can get information about what were the circumstances surrounding the episode and what happened after the episode. And through that, we can, in a very short space of time, get some indication of whether this is a true seizure or not, and get some feedback. So for those who are, we're hoping that this is going to be rolled out more internationally, but for right now, it's currently being done in the UK. And in addition to the videos, we've started looking at artificial intelligence to see if we can apply that so that it can be used in other places, such as in low middle income countries as well. So we spoke about the seizure types, you know, how they look, but also how they start and where they are in the body. So they could have a focal onset, or it could be a more generalized onset. And within that, we want to know what are the potential causes. So is it structural in nature? Is it genetic? Could it be infectious in nature or metabolic in nature? And from this, we can put together the epilepsy type based on the seizure type. So if it's just focal seizures, then we will say that's a focal epilepsy. If it's generalized onset, then we say that it's a generalized epilepsy. But some people can have a combination of different types of seizures as well. And sometimes we don't know how the seizures start based on the history, or we haven't had an EEG yet. And so we don't know exactly what's the epilepsy type. Sometimes putting all this together, we can formulate exactly what's an epilepsy syndrome, and that way we can get a better prognosis and indication as to what's the likely outcomes. And related to that, we can talk about the additional problems aside from the seizures. So we're very interested and very keen to have a holistic approach and make sure we look at cognitive, behavioral, gastrointestinal, and sleep problems as well. So this is the latest classification that is used. So we talk about focal seizures or generalized seizures, and within that, we talk about motor seizures, which you can see actual movement and or not, and if there is loss of awareness or there is preservation of awareness. So these are terms I think that some of you might be familiar with. We no longer tend to use this in the medical aspect, but we know what they mean. And so people talk about complex partial seizures, simple partial seizures. So a simple partial seizure in the new currency would be a focal seizure in which there is consciousness is preserved. And a partial seizure is a focal seizure, and so on. And if you're speaking with much older people, they might refer to it as petit mal seizures or grand mal seizures, referring to tonic-clonic seizures in the case of grand mal seizures. So we talk about epilepsy syndromes, we can talk about age of onset, and you will hear this particular term used a lot, developmental and epileptic encephalopathy. And arguably, EEF1A2 is a developmental and epileptic encephalopathy in which the underlying etiology, in this case, EEF1A2 mutations, plus the epilepsy activity itself, contribute to the developmental problem. So the better you can control the seizures, then the better the outlook would be. But even when you control the seizures, there will still be an underlying problem, because the developmental etiology contributes to the actual impairment itself. We've done how we make diagnosis, a combination of the history, physical examination, and we do some tests, but largely the history it is the most important thing. So a video and knowing what happens before and what happens afterwards is crucial. And in terms of tests, the main test that we tend to do to try to make the diagnosis is an EEG. As I mentioned before, a video is incredibly helpful. So in terms of EEG going forward, so how we normally examine an EEG is that we have people like myself or neurophysiologists who are experienced in looking at EEGs. And then when we look at EEGs, there are particular patterns that we look for in order to try to make that diagnosis or not. And sometimes there are different types of EEGs. Sometimes in a standard EEG, which is 30 minutes long, sometimes we don't see a seizure during those, and the EEG looks completely normal, but that doesn't rule it out. So the ideal thing is to try to capture an event and to see if there are electrical changes as you go along as well. Needless to say, that's quite labor intensive. So there have now been AI and machine learning tools in order to try to say if EEG is diagnostic or not. And this is just an example of a prospective study that is going on to look at that as well. Some children who have epilepsy have behavioral problems, including autism. And so it might be very difficult to have lots of leads on as well. So we've been looking at whether we can reduce the number of leads, and we are having some very promising data looking at just 4 electrode placements that can help with that diagnosis as well. What about treatments? Well, there is a long list of medications that can be used, and this is very UK-centric. It's in the National Institute of Clinical Excellence and Scottish Intercollegiate guidelines. And it's really based on the seizure type. So we tend to choose medication based on the seizure type, whether it tends to be more generalized or focal in terms of its onset, and also what they look like. Are they tonic-clonic? Are they myoclonic in nature at all? So you can see there's actually quite a bit of overlap between the medications that are recommended for generalized seizures and focal seizures. So what's probably more important are the medications that are not to be used for a particular condition. In the case of EEF1A2, regrettably, we don't yet have a very specific set of medications that work extremely well for EEF1A2. So there are other syndromes in which we seem to have a better understanding. And so, for example, in Dravet syndrome, which is developmental and epileptic encephalopathy, is one of the severe early onset epilepsies. We know that there are some drugs that are much more effective in those compared to others. Lots of the medications have multiple different mechanisms or purported mechanisms of action. So you see, for example, in topiramate here, you see it's a number of different ways. So I think all of these approaches are really treating the seizures, and they're anti-seizure medications rather than anti-epileptic medications. And I think there is a real need for anti-epileptic medications and precision medication just to address the specific abnormalities. And you would have heard a lot of the stuff that Kathy and her group are doing in order to address that. Kathy touched a bit on gene therapies, and she's got this award to look at the ASO therapy, looking at that. There's some really promising results from that in the Dravet syndromes in terms of repurpose. It's not repurposing as such, but it's traveling down a path which is already being laid, and we've seen that being laid for Dravet syndrome, and really promising results regarding that as well. So what about the future? Some people have been asking, are there particular patterns that one can predict? So some people ask about during puberty, does that make a difference? During particular menstrual cycles, does that also make a difference? So another tip is to use a seizure diary. So if you can use this app, which is called the SEER app, which is freely downloadable. And within that SEER app, you can annotate and record the seizures, but you can also record other things such as mood, sleep pattern, and things like that as well. And this is some nice work which was done by Carolee here. And what they did is that they used the SEER app, and they had self-reported seizure types. And from this, over a period of time, they were able to adapt and see what the patterns were like for individuals. And from this, they were able to estimate a seizure likelihood. So you get some idea, am I going to have a bad seizure day, or am I going to have a good seizure day? So it's a bit like weather forecasting, but specifically for seizures. And with this, you can make alternative or make necessary preparations in order to try to minimize your risk. This is some work by Andrea Biondi, again, seizure forecasting, and this is using the SEER app as well as Fitbit. And so they're able to look at heart rate, look at sleep, look at stress patterns, and looking at mood, and they were able to get an idea of the risk for individuals. So what you'll see here is that these marks here are specifically related, the red marks are specifically related to seizures, and they could see and predict for some individuals that they could predict when a seizure was going to occur based on previous history, according to combining their heart rate characteristics, sleep characteristics, and stress characteristics as well. So I'll just end there. We don't yet have a specific treatment. A number of different treatments work. Levetiracetam and valproate and clobazam are typically some of the more commonly used medications because the initial presentation is myoclonic seizures. But I'm afraid we don't yet have a golden bullet, and but a lot of people are working very hard at that. Thank you very much.
Oh, thank you so much, Richard. So up next, we have a talk from Dr. Lindsay Mizzen. She is an honorary senior clinical research fellow in the Patrick Wild Centre at the University of Edinburgh. Her main interest is sleep and single gene conditions that are associated with intellectual disability. But she's also an NHS consultant psychiatrist who works with adults with intellectual disability. So Lindsay, you can go ahead and share your screen. And
thank you very much for inviting me, Heather. Here is something completely different, just to give you an idea of what can happen in genetic conditions in terms of other sorts of research. So the condition that I have been researching is called SYNGAP1 related intellectual disability. I'm just going to call it SYNGAP1 so it's not so much of a mouthful. It's another condition that is caused by changes in a single gene. It's not changing. There we are. And this is the SYNGAP gene. So it's a gene that codes for a protein called SYNGAP1, and that is found in the brain at synapses, which are the junctions between different neurons, different brain cells. It's a protein that interacts with many, many other proteins, and it has a role in regulating the transmission of signals from one brain cell to the next. In people who are affected by changes in the SYNGAP gene and therefore have SYNGAP1 related intellectual disability, they often have intellectual disability that is moderate to severe, although I just have a sense that we're now starting to identify people who have a bit more of a milder phenotype, so milder presentation, which is probably because people are being tested in terms of their genetics for a wider range of presentations. At the moment, at the end of 2023, the official census recorded by the SYNGAP Research Fund, which is a large family support network and research funder for SYNGAP, their census showed that there were 1,339 people worldwide who had signed up to say, I have a diagnosis. We know there will be many more people than that because there'll be people who haven't opted into that, but also, as has already been said, people who are older, so perhaps older children, but certainly adults are less likely to have been offered genetic testing in the past and therefore may well have SYNGAP without knowing it. So my research is centered around sleep. This was mainly because looking at the research papers for SYNGAP, you would have a list of symptoms that people were suffering from, and sleep problems were highly prevalent, but nobody had sort of systematically looked at that yet. So before we actually look at the research, we need to define what we're talking about. So we all know what sleep is, but actually when I sat down to write the talk, I realized I couldn't give you a definite sort of description of what it is. So the Cambridge Dictionary tells us that sleep is the resting state in which the body is not active and the mind is unconscious. Now, obviously, we spend a significant amount of our lives in sleep, some more than others. So it must be important. What does it do? Well, we know that if sleep is disrupted or the sleep-wake cycle, the circadian rhythm is disrupted, it can have all sorts of effects, and we know what it feels like, all of us, but specifically things like concentration, mood, ability to learn, general health, and also it can impair your immune function and make you more susceptible to things like infections. So I wanted to look at whether you could find biomarkers, so sort of specific patterns in SYNGAP1 sleep that you couldn't identify in people without that condition. And I will come back to the fact that I was thinking about this cross species a bit later on, but it's because I have collaborated with Dr. Alfredo Gonzalez Sousa, who is also at the University of Edinburgh, who was looking at SYNGAP1 rats and how well they sleep. So what did we do? We had a variety of different things. So we sent out questionnaires to participants across the UK. They included medical information, medication, development, social information, so the children's sleep habits questionnaire, and a questionnaire about quality of life. We asked participants to wear a actigraphy watch for 7 days. So that's a bit like a Fitbit. It You wear it literally like a watch. It is just a Velcro strap, and it measures levels of activity over the course of that week. So it gives you a proxy for when somebody's asleep and when they're awake. We asked people to complete a 7-day sleep diary, and we then did a 2-night sleep recording in the child's own home. And I've just realised that putting Stevie Williams on the slide, I still didn't mention him at the start. I moved him from the end because I tend to do that. So just to come back to that, sorry, Stevie Williams was my research assistant on this project. And it was him that went around the UK to the homes of families affected by SYNGAP1 to do the recordings in their own environment. The equipment you can see here, I don't know if you can see my pointer, but in the middle at the top, we have 2 participants who were wearing the kit. So it is all attached to the person. It all comes around into a sort of little box here that just sits just under your breastbone, and it measures all sorts of elements, which I will come to in a minute. It's just picking out bits that might be interesting for you from our results. We recruited 15 children with SYNGAP1 and 15 children without, who we call our control group. And in terms of the children's sleep habits questionnaire, we totted up all the scores and found that their score is worse sleep. And typically a score of over 41 indicates poor sleep. So you can see that the SYNGAP1 group had a score of 51.6 on average. You will also notice, if you look carefully, that the control group actually had an average of 42. So they alone could be described as having sleep problems. We don't necessarily understand why that might be, but it could be because having a sibling with SYNGAP1 might be disruptive in the household. And we do know that some of the participants co-slept with their sibling, because typically we tried to have somebody who was a sibling of the affected individual, so that when we were recording in the same house, it meant that there weren't so many environmental differences. So say a fire alarm went off, then it would have affected both of them and it wouldn't have skewed the results quite as much. So in terms of those sleep recordings, we measured EEG. So we've heard about this today already. So measuring the brain waves, the brain activity. And then to score different types of sleep, you need other measures. So there's measures of eye movement, oxygen levels, video, and the airflow coming and going as somebody breathes. We actually had to give up on that aspect because children nose there, but we were able to record pulse. Underneath, I'm not going to go into this in any detail just from a time point of view, but I thought I would just show you what's a sort of, we call this a hypnogram, what it looks like when you put a whole night's sleep recording together. So you may well be aware that there are different stages of sleep. And in this graph, if you're at the top level, then you're awake. And then as it goes down, right down at the bottom, we're in the deepest sleep. And in between, you have different types of sleep. The red lines are indicative of REM sleep, so rapid eye movement sleep. That colloquially tends to be referred to as dream sleep. It's not the only part of your sleep in which you dream, but dreams are particularly vivid during that period. So what did we find? Well, we discovered that although most of the sleep stages, so the sleep tends to be split into 2 main stages. You have non-REM sleep, and then you have the REM sleep that I've just described. The non-REM is split into 3 different sets of sleep. When we looked at those 4 different stages of sleep, the only one that was significantly different between the people with SYNGAP and our control group was the REM sleep. So there's something going on in specific aspects of sleep. what's really important about this, and comes back to what I was saying in terms of what we were hoping to achieve in this project, is that it is exactly the same finding as Dr. Gonzalez Sulcer found in rat models of SYNGAP. Now that is important, because if it means that tests are ongoing in the animals for potential therapies, then we can use the same technique that they use, so the EEG brain activity recording, in people. And therefore, if we were to test the same medicines at some point, then we could directly compare across and see whether there is a response to treatment. So I've already mentioned this really, but just quickly, in terms of the rapid eye movement sleep, during this phase, your brain activity increases, you get temporary muscle paralysis, except your eyes and obviously your breathing. So this is why we look at eye movement as part of the sleep recordings. It's a very important sleep phase for memory, learning and creativity, and has the most vivid dreams. So clearly, if you're not having as much REM sleep, then this is going to impact on your daily function, potentially in terms of memory and learning, that sort of thing. So I just want to come briefly to the epilepsy side of things in SYNGAP, because it is frequently reported in some of the research papers. Everybody's diagnosed with epilepsy. In our situation, 8 of the 13 participants that we got successful sleep data on, so as I said before, we had 15 people with SYNGAP recruited. We got 13 people we had sufficient sleep data to analyse, and 8 of those in the SYNGAP group had a diagnosis of epilepsy. None of the control group had epilepsy. Out of those 8, only 3 had recordings on our sleep data that could be indicative of seizures. There was somebody else that we know from sleep diary was having absent seizures during the day, but nothing was seen overnight. So we had 3 people with episodes that looked like they could be seizures. Again, this is important because of the rat data. So in the rats, as I've tried to show here on the left, there was evidence of absent seizures in quiet wakefulness. So when the rats were going to sleep or waking up. And hopefully you can see my pointer, because I'm just showing you here in the middle of the SYNGAP rat panel here, there's this change in all these brain waves that shows that the rat is having a seizure at that point. And then hopefully you can see on the right that in the top panel in particular, it's really quite similar. There's this point in the middle of the recording where it looks very much like a seizure. Some of our recordings, we did have confirmation later from the participants' neurologist that they thought it was seizures, or the parents reported back. But for others, it was our interpretation. Stevie, who did the analysis, is a very experienced sleep physiologist, but neither of us are neurologists. So we weren't able to offer the family any diagnostic information, but if we saw something unusual, we provided them with the data to share with the doctors. And just at the bottom here is another change in the recording that is indicative, we think, of seizures. So that's as far as we've got with our sleep research at the moment. We are still writing up the data and looking at various other aspects. But I hope that's been interesting. There are people I know on the call who, or potentially on the call, who may remember that I was actually in touch with the EEF 1A2 family group last year, because Kathy and I had talked about doing a study where we could compare sleep in EEF 1A2 in SYNGAP and also a third genetic condition. Unfortunately, we weren't successful in getting that funding, but I continue to look out for opportunities, and it may be that we can revisit that sort of idea at some point in the future. So thank you for having me.
Thank you so much, Lindsay. So next we were supposed to be hearing from Izzy Utley. Unfortunately, she isn't able to be with us tonight, but she's very kindly recorded her talk. So I'm going to have a go at sharing that recording. Izzy is a speech and language therapist. She currently works in the National Autism Implementation Team, and she works clinically in the Adult Learning Disability Service. And she has previous experience in Children's Learning Disability Services.
Hi everybody, good evening, good morning. I know people are watching this in different areas. Sorry that I can't be with you live tonight. I'm hoping this video has made its way to you. My name's Izzy Utley. I'm a speech and language therapist in Scotland in the UK, also a research fellow at Queen Margaret University, and I'm going to do a quick whistle-stop brief tour of different communication strategies and environments. So the plan for sort of the next 15 minutes is to mainly try and focus on these things. So thinking about what do we mean by communication, some simple strategies to make good communication environments, and some signposting to some other resources and other strategies that you can use. So I think just to start with, just to say that obviously communication can be completely different for different people. All of your children have probably got very unique communication profiles and preferences. I'm not going to be able to cover everything for every individual today. We're going to take quite a broad view. But I guess just to highlight that if you have got concerns about communication or you want further communication support, then a speech and language therapist is a good person to get in contact with. So in the UK, we're speech and language therapists. Across in the US and in other areas, we might be known as speech and language pathologists. There might be other professionals as well who might be supporting your child who can help with communication, so particularly kind of early years staff or school staff. So please do get in touch if you would like with them, if you would like more individualised support. I'm hoping, again, I know some of you, a lot of this might not be new to you, you might know quite a lot. If so, just, I guess, sit back, enjoy a cup of tea, enjoy. For other people, obviously, this might be something which is quite new, and please do take the time just to all of the information I'm giving, there's links to different things. Don't expect you to kind of know all of this now, just take your time kind of going through things. So the first thing, I guess, to highlight about communication, clearly, obviously, it involves more than one person, but actually that's something that I think we sometimes forget and something that's actually really important, and why we talk about changing communication environments and not just changing the communication of the person who's maybe having difficulties. Actually, we've got a joint responsibility for communication, so all of us need to change our communication so that we're supporting people, and that includes anybody that is supporting your family, so any of the medical team, any of your early years teams, school teams, adult social care teams, that everybody has this responsibility. And when we talk about communication, really crudely, we kind of talk about expressive and receptive language. So expressive communication is where you're trying to communicate your message to somebody else, and receptive is where you are understanding the other person's message. And it's quite important to kind of have a differentiation between these, just because people can have different skills in different areas. So sometimes we get people where actually their expressive and their receptive skills are perhaps at the same level, and sometimes we get people who perhaps have really good expressive communication but perhaps don't understand as much, or it can be the other way around, where someone might be able to understand a lot but they don't have any spoken communication, or the expressive communication is really difficult for them. And this is where it's really important that you obviously know your own child best, and we think about supporting everyone individually. And the other thing just to highlight is that obviously we're seeing this within the context of the whole person. So previously, I think we've taken communication away and we've kind of put it in a little box and tried to work on it. And actually, really, we need to be thinking about all of these things which can impact on communication as well. So particularly, I guess in this diagram, thinking about socioeconomic status, nationality, race, culture, all of that can have an impact on how somebody communicates. And actually, it's really important that we keep these things about the whole person, that we don't try and take communication away from them. So just to go through a bit about the, I guess, kind of the theory around communication. This is quite an old diagram, but I quite like it still. It kind of shows the 3 different things that really we need to have successful communication. So we need a means, which is how we communicate. We need a reason, which is why we're communicating. And then we need some opportunities. So we need, opportunities to communicate. So if I go through each one, so reasons, obviously there's a huge range and this isn't covering all of them, but I think it's quite key to remember that there are loads of reasons for communicating, and often we can get stuck on the kind of the idea of requesting and choice making with somebody. And actually it's really key that we remember that all of these might be things that people might do, sort of reasons why they might want to communicate. And I guess making sure that we're meeting those needs as well. So then we've got a range of means of communicating, and as a society, we tend to favour speech, and that seems to be the thing that most people use to communicate. We might use signing as well, but actually there are a whole range of means of communicating, and the importance actually is that there isn't one way that's necessarily that we should be thinking of as best or superior. Actually, all of these are good ways of communicating, and if someone communicates in a different way, that's okay. So you might have, your child might communicate through their body language, or they might make slightly different intonation, they might make slightly different sounds for different things that you can understand, and that's their way of communicating. And then we need opportunities. So perhaps these are a bit more adult focused, but I guess people need something to communicate about and an opportunity to communicate. So particularly thinking about, have people got a chance to form relationships? You could have the means and you could have the reason that you want to form relationships and friendships, but actually if you're not going out and meeting other people, then that might not happen. So the opportunity for this to take place is really key as well. So it's just thinking about having all of those things present. And then the other thing, just to briefly think about, is what does it feel like when we can't communicate? And one of the things that I quite often think with this, for me, I know it's been difficult when I've perhaps been away somewhere and I've not spoken the language of the country that I've been in, or it hasn't been my first language. And actually I can then think, actually I've felt all of these things before, really frustrated that I can't get my message across, that I can't get what I want, really tired, which again, if you're then thinking about particularly your children who might be, any children that might be having seizures and other things which are affecting their tiredness, actually if communicating is really difficult for you, that can be really tiring as well. And actually it can change your personality. So if you are quite an outgoing person, but then you go somewhere or you are in a situation where you can't communicate, that can really change who you are as a person. So I've got I'm halfway through. I'm going to do another kind of 7-8 minutes and just think about some of the specific approaches that we can use. This is not by any means an exhaustive list of approaches. It's just a few ideas that might be useful and might kind of signpost you of things to look at. And I'm going to go through each one briefly. So intensive interaction is a way of interacting with someone. It was based on kind of parent-child interaction, and I guess the thing to think about is that it is Western style parent-child interaction, so it might not be suitable or of interest to everybody. It's designed for people at quite early stages of development, and it supports development of relationships and interactions. And probably the best thing, I could do training on a whole hour's training just on intensive interaction. The best thing to do is to visit their website. They've got loads of videos on there, and they offer training and further information. I would say if you're in the UK, if you're in a special educational needs setting, they would probably know about intensive interaction. But it's a great approach for supporting people. Then we could have a look at AAC. So this is alternative and augmentative communication. And we have two different types of AAC. So you can have high tech and you can have low tech. High tech AAC just basically means it's got a dynamic screen or some kind of voice output. So something like your apps on your iPad or one of the switches that maybe has quite a lot of buttons that you can press. And low tech is more the paper-based stuff. And there isn't really, there's pros and cons for both of these. High tech, obviously, if you've got something on a dynamic screen, then that's great because you can have loads of different visuals. But equally, if the battery suddenly dies, then, your low tech is really good as well. And these are the things that are probably really key, that they're very individualized for each person. So some people won't cope with different types of AAC, and also if you've got children that have got physical difficulties, they might need different ways to access things. And there's so many ways that you can access different things now, different switches, different positions. Occupational therapists can be really useful at looking at kind of positioning and support for AAC devices, or even paper-based AAC. So there's a couple of just examples on the screen here of a sort of a device that's got some symbols on, and then something called a now and next, which is just showing the person what's happening now and what's happening next. Just to briefly touch on signing systems. So again, this is maybe quite UK centric, but signing systems are different. So they're different to kind of BSL, British Sign Language, or American Sign Language. So BSL and ASL are kind of their languages within their own rights. They've got their own syntax and vocabulary. What I'm talking about here are systems where they're called sign supported systems. So Makaton and Sign Along in the UK, and that it's about signing keywords. And signing can be a really useful thing because similarly to using visual things, it just helps it helps people to process the message. So if you're just speaking to somebody, it can be quite hard for them to understand and to process. Whereas actually, if you're signing as well, that can be really helpful to some people. In terms of which signing system, it completely varies wherever you are, and probably the best thing to do would be to find out what your local area, which kind of signing system they use, and they might be able to offer free training on this. It's quite often used within kind of special schools, and they'll follow kind of one of these. In the UK, probably one of these two options. Then talking mats, I'm going to just mention this very briefly because I think this is something where the person who's using it probably needs quite a lot of skills to think about a question hypothetically, but it can be a visual tool for helping people to express their feelings and give opinions. And I've put the website there. So again, similarly to the intensive interaction, there's quite a lot of videos, I think, possibly on the website, or if you do a search, you can find videos of how it works. Communication passports are really key. So this is a way to pass on key information about a person when they can't communicate it for themselves. So there's a link here, and I think there's a couple of blank communication passport templates that you can see. And this is a really key thing, particularly for people that are moving into different settings. So perhaps if your child's starting school or if they're moving between classes, having a passport which has got the main information about them, and I guess the ideal is for the person to communicate, to design the communication passport with you, and think about, what pictures do they want in there, what do they want someone to know about them, and I guess just to highlight that actually there is movement in developing more digital communication passports. So again, you might have children who perhaps they, like I mentioned earlier, perhaps they change their intonation as a way of communicating, and actually having a digital passport is brilliant because you can have a video of something that might be quite hard to describe in pictures and words, and you can have this video kind of embedded within the passport so that people can see what it's like. If you, I mean, I assume there'll be quite a few people, but PAMIS in Scotland is one organisation that's been using this, but there may be others around. And I guess it's just something, if you're aware of communications passports, just to be aware of, that is quite new. And then finally, objects of reference. So these can be used to help people understand what's happening next. And it's just using an object to give someone a clue of what's happening. And you can have a specified amount of objects, and you can set this up with your speech and language therapist. But actually, it's probably something that you're doing at home anyway. So for example, if you're showing a towel before bath time, or perhaps getting car keys before you go out, and letting your child kind of look at the car keys or feel them if it's appropriate, that can be really helpful with letting somebody know what's about to happen. So it can be useful to think about, are there things that you're already doing, or things that, you know, perhaps if there's difficult times, you know, giving someone a spoon just before breakfast so that they get to feel the spoon and they can then understand that breakfast is coming. So I feel like I've done kind of a whistle-stop tour through a few of the more specific approaches. And just to finish up, just kind of a few, I guess, tips of how we can support good communication. So you might hear about a total communication environment. So that just means that we're using any type of communication. So you might have, for your child, there might be some signing going on. There might be some, you might also be using some symbols and some photos. You might also be watching their body language and their facial expressions to see what's going on. And the thing about total communication environment is that we're respecting all types of communication. So it doesn't matter the way that the person is communicating. It's just important that we're following that communication and honouring that communication. The really key thing is about knowing the person well. So obviously, as the parents, you are the experts. You're the ones that can fill in the communication passport and that can advise other people. Reducing other environment stressors is really important. So probably people will have experiences of, you know, if you've been driving and one of your children's screaming in the back, it can be really hard to concentrate and actually have a conversation. So that is true for all of our children as well, that actually if there's other things going on, if they've got sensory sensitivities or, you know, particular places they don't like, communication is going to be more difficult in those settings. And then just whizzing through the next three because I'm running out of time, but following lead and pace, using simple language and checking they've understood if that's appropriate, and using what's in your environment and being creative. So again, really using kind of objects that you've already got in the house, or even using drawing as well. I think sometimes having symbols people can think, oh no, I can't do this because I haven't printed out symbols, I haven't laminated them, it's not perfect. But actually just be creative and use drawing and writing and anything else that you've got. I always think communication is just a bit like detective work, and you're trying to kind of get through and get the message together, and so there's lots of different ways of doing this. So that's me done. As I said, sorry, slight whistle-stop tour. If you've got any questions, please do let Heather or the team know. I'd be happy to answer those. And again, sorry that I can't join you this evening. Thank you.
So we're just going to go through some of the questions that have been put into the Q&A, and hopefully we should be able to see and hear everyone who's answering the questions. So just to start off with, we'll maybe start off with the timescale question. So one of the questions that came up a few times was what sort of timescale we're looking at for these therapies that we are maybe talked about in the future, maybe in terms of years.
Yeah, I mean, it's a very hard question to answer because even when things are going well in the lab, which they are with the antisense oligonucleotides and Tamsin starting the drug repurposing screens, there's, even once you've got to the point where you've done testing in animal models and things, and we're not even there yet, you've then got a lot to get through in terms of sort of regulatory authority and things for setting up trials. The challenge is a rare disorder are really quite substantial. I certainly get the impression that things are better than they were. There are things like the N. Lorem Foundation in the US, which specialise in treating sort of N of 1, but actually getting the evidence required to get the approvals from the FDA and things like that is a very time-consuming task. So people usually talk in terms of 5 to 10 years, but it can be a lot longer than that. And I think we just have to wait and see. I'm really sorry. I'd love to be able to give a more definitive answer, but it will partly depend on the type of therapy that becomes available, because repurposed drugs, as Tamsin said, can get into the clinic much more quickly than any kind of genetic-based therapy.
Thanks, Kathy. So, sorry. One of the questions which I've lost. No. Sorry, I just can't find the exact question I wanted to look at. So Lindsay already answered this question about her own work in SYNGAP 1, but Catherine asked, thank you for these talks. How did you become interested in this specific gene? And for any gene therapy in the future, would this depend on each variant?
Shall I do that again?
Sorry, Kathy, and then I'll
do another.
Yes,
Yeah. Been doing it for longer. I actually have been, well, as you saw, I've been working on EF1A2 for over 25 years. I got interested because there was a particular mouse strain that had a very interesting neurological phenotype, and I was trying to identify the gene that caused it. This was in the days before the human genome had been sequenced, so it was a sort of long, laborious task, and the gene turned out to be EF1A2, and I've been working on it ever since. And then, as you heard, about 15 years later, the first patient was described with missense mutations in EF1A2, and that was the first time we knew what the results would be like in humans. So we kind of switched much more to working on EF1A2 related neurodevelopmental disorder. And that's a very good question about the gene therapy. It depends exactly what type of genetic therapy we're talking about. So if you were going to do gene therapy where you were putting the gene back in an AAV construct or something, the way it's working so well with Rett syndrome that you'll hear about from Monica, then that would not be dependent on the mutation. But for things like the antisense oligonucleotides, they have to be redesigned specifically for every single mutation. And of course, that also means you've got to have in an ideal world, you've got to have the mouse models to test them on. So drug repurposing, we would probably be able to group mutations and get an idea about loss and gain of function. So it wouldn't be quite as laborious as with the ASOs, but it's I think Richard said this in one of the answers, it's sort of how long's a piece of string, because until we've got a therapy that we can then start developing, we won't know whether it's got to be done individually in that way. Oh, sorry.
Sorry. Sorry. Okay. So this may be one for Richard. Oh no, it's just.
Do you want me to read one out for Richard?
Yeah. Please, thank you. I'm going to jump on it.
So, Richard, there's one of the most recent questions is, how common is it for seizure activity to have a pattern, like, for example, happening every 10 days, 4 days, etc.? Because this person who's anonymous says that his son had seizures every 2 weeks in the first 5 months, always on time, could set his or her watch to them. Oddly enough, when he turned 6 months old, they magically went away without medicine.
Yeah, it's actually more common than people actually realize, and it's the basis on which people have been doing the seizure forecasting. So what people have looked at is to look at the seizure patterns over usually a 12-week period and extrapolate from that to see where the patterns are. And there are some people who, for example, only have seizures around the time of their period. There are some people who only have seizures at nighttime. So there are circadian rhythms and there are hormonal rhythms that can necessitate and contribute to that process. So it's more common than people actually realize.
Thank you. Heather is still choking, I'm afraid, so I'm going to just keep going. So Keely has just asked, is there a potential for EF1A2 gene therapy to improve quality of life in other areas of learning and development apart from epilepsy? For example, the potential for a non-verbal individual to learn to speak. We would certainly hope so. I mean, that's really the idea of the genetic therapies, is to try and get at the real underlying condition. Monica, I don't know if you would feel able to chip in on this.
I'm sorry, can you repeat the question, Kathy?
Yeah, is there potential for gene therapy to improve quality of life in other areas of learning and development apart from epilepsy? For example, the potential for a non-verbal individual to learn to speak.
Well, that's the hope. I think we'll have to, we'll have to wait and see. In our case, the gene therapy trials just started. And there's been a total, I think, of 6 patients that have been dosed. So we'll have to see. The hope is that the improvements will be broad and spread over many symptoms, but we don't know yet.
Great, thank you. There's also a question, are there any prospects for supplementing EF1A2 exogenously? And I think there was an earlier question about whether we could replace the protein. I think the real issue with putting the protein in is, and with a lot of things like this, is actually accessing the right tissues. That there's been work with enzyme replacement therapy in things like ADA deficiency, where you are delivering the protein, but then it's to the blood. It's very, very different trying to deliver something to the brain. And proteins themselves are digested in the gut. So, actually getting something directly to the tissue affected is often the biggest challenge, really, with that. And I don't think we're anywhere near that point, unfortunately. Is there any research on the benefits of deep brain stimulation for epilepsy? Richard, would you be able to come in there?
Yeah, there's certainly work looking at that deep brain stimulation, and people are looking at different target places. So the thalamus is one particular place, and there is a big trial that had been done in the States about that. People have been trying to look at other areas as well, and people have been looking at where the optogenetics would play a role, but there's certainly work on deep brain stimulation, but nothing specific about EF1A2 that I'm aware of.
Thank you. Sorry. Now you don't know which questions we've covered them. So we've talked about timescales, and somebody asked about updates on the gene therapy that downregulates the mutant form of EF1A2. I don't know if you want to say anything about.
Yeah, so that's exactly what I'm currently trying to achieve with antisense oligonucleotides. So I'm focused at the minute on E122K, but we seem to be having some success at this point in cells, specifically knocking down E122K with, depending on the ASO that we use, mild or very little impact on the wild type protein, the wild type transcript. So if that continues to be successful, then we'll start moving that into the mouse models to see it has benefit against their phenotypes.
Thank you. And I think somebody, I can't see all the questions because I moved computers halfway through, but somebody asked about the D252H mutation, I think, before I left my office. And I was just going to say we've not done quite as much work on that, but we have a master student starting next week, Nandini, who's going to work with Grant on ASOs for that variant as well. Lindsay, has got a hand up.
I just wanted to come back in on the question about whether gene therapy will lead to sustained improvement in various other aspects of life. In terms of SYNGAP, we're also not at a stage of having any genetic therapy, on the horizon right now. But what we're hoping is that some of the sleep research, we hope if that can then lead to improvements in sleep, that will lead to improvements in daily functioning. And we don't know yet just how that might help over the developmental life course. But I suppose I just wanted to put it out there that ideally everybody would have a gene therapy that corrected the underlying condition, but we're hoping that there are other ways around this to make improvements for people even whilst we're waiting for that kind of therapy.
Thank you.
Sorry, I'm not sure what's being asked. I don't know if you just want to keep going with it. There's a few questions to chat in the Q&A session.
Oh yeah. Corey, that's a very good question. Is it likely this condition will be renamed in the future, more of a common name, or is it too rare? This is something that comes up quite a lot with these, particularly with these sort of rare epilepsies, because it's just alphabet soup, isn't it? And I don't know of, maybe Richard will be able to speak to this, but I don't know of any move towards renaming anything. It's a complete mouthful, but I think it is what it is, and we're probably stuck with it, I'm afraid.
If anything, it will be the other way around. People have been trying to say to keep away from naming particular conditions after particular groups of people, such as Dravet or Lennox-Gastaut and so on, because it doesn't really tell you about the disease, and two, it suggests an ownership which people are trying to get away from.
Thank you. And what's the best way to share information with the research team about medications that are working well or failed medications for specific variants? That's another really good question. Richard and I put an enormous amount of effort into setting up a registry for EF182 that many of you have contributed to. And it was being held by what was then called Patient Crossroads, that was taken over by Invitae. Invitae have kind of pulled out of that. They sacked all the staff, basically. Then it suddenly came back again, and I just don't know what's happening with it. I got quite a few emails from people from companies wanting to take over the registry, but the costs are astronomical. And again, this is something that Monica can speak to, but it may preempt your talk, because there are possible ways around this in the future. But at the moment, it really has to be something like that. I mean, it's super appreciated when people send us information, but I can't use it systematically. We don't have the ethics to be able to do much with it, really. So a registry that all researchers can access and all clinicians can access will be the way to go. But thank you.
Kathy, just a question. Was the registry at Invitae, was that part of Citizen, or was it, do you know? Citizen started off being independent, then was bought by Invitae, but has since spun off again and is independent again. That's a digital natural history study where they extract information from medical records. Is that what you're talking about?
No, it's not. We set it up ourselves with they have some basic questionnaires. It was called Patient Crossroads at the time. And we used their basic questionnaires, and then we wrote specific ones for EF1A2.
And Invitae bought Patient Crossroads?
Yeah.
Okay.
This is quite a
All right,
while.
well, I do have some suggestions, which I'll touch on when I'm giving my talk.
Thank you.
We'll maybe do one more, one more, sorry.
Is there any research linking gastrointestinal problems and autoimmune issues? Yeah, so the short answer is no. I know a lot of you have problems with this. I don't know if Richard will be able to say any more. My assumption, because EF1A2 is not found in gut cells per se, but obviously the gut has to be innervated to be able to work properly. So the nervous system has to work. And there are nerves in the gut which will be expressing EF1A2. So I assume it's a sort of secondary thing, but I don't know.
I'm afraid I don't know either, Kathy. I would be speculating, and I'll do speculating along similar lines, because we know that some children who have a dystonic type of cerebral palsy, they also have dystonic gut. So it's not uncommon for children with neurological problems to have gut problems, and it's particularly those who have movement problems.
Thank you. I'll maybe just very quickly answer that. There's a question about the lifespan of the EF1A2 mice, and just because this came up before, and just to say, when we're talking about the mice that you could see on Grant's figure, they were homozygous. So these were mice that had no EF1A2 at all. All of your children have a normal copy. And the mice that have one normal copy have a completely normal lifespan. There isn't any effect on survival if you've got one copy.
Thank you very much, everyone. Sorry for jumping out in the middle. But up next we have a talk from Monica Conrads. She is the founder and the CEO of the Rett Syndrome Research Trust, and she is the mother of an adult daughter with Rett syndrome. And she's going to talk to us about a patient advocacy journey. So you can go ahead, Monica, and I'll just.
Great, thank you, Heather, and thank you, Kathy, for inviting me. Okay, let me just. Okay, so as I was listening to the introductory talks from Heather and Kathy, there is quite a bit of overlap between our disorders and probably a lot of shared learnings as well. So Rett syndrome affects about 1 in 10,000 females. It's almost exclusively diagnosed in girls. There's a genetic reason for that I won't go into, but in rare cases, it is also found in boys. The gene that's affected is called MECP2. It's on the X chromosome. Girls have 2 X's. One of them is silent. And so a girl with Rett syndrome has about 50% of her cells that have the mutated protein, and the other 50% that have normal protein. That gives you full-blown Rett syndrome. In the case of the boys, they don't have that backup protein. So they have no MECP2 or no functioning MECP2. And so they're typically much more severe. There's no at-risk population. You find the same incidence regardless of ethnicity or race or any kind of socioeconomic factors. It is mutations in a single gene, which is good because it means we have a clear target for intervention. There's been about 900 mutations that have been identified, although there's about a dozen or so that encompass about 50% of Rett cases. It affects nearly every symptom in the body, and I'll use my daughter as an example. She's 27. She can't speak, she can't walk. She has intractable seizures fed through a feeding tube. cannot use her hands at all. In fact, loss of purposeful hand use replaced by a stereotypic movement is the hallmark symptom of Rett. She also has problems with sleep, breathing irregularities, breath holding and hyperventilation, and anxiety, scoliosis. In nearly 100% of the patients, about a third undergo scoliosis surgery. So it's a very severe disorder. The kids grow up to be adults and can live many decades, but quality of life is, pretty hampered. It's not neurodegenerative, which is good news. That means that the brain cells are not dying. And we do not have any effective treatments. Our kids, like your kids, are on sleep medications, antidepressants, anxiety meds, seizure medications, GI. Oh, and I didn't mention GI. They have a lot of GI issues as well. The gene was discovered by Kathy's husband, Adrian Bird, in the early 90s. And then in 1999, it was discovered by Huda Zogby at Baylor in Houston that mutations in MECP2 were causing Rett syndrome. That was around the time that I became involved. My daughter was diagnosed in 1998. So a year later, the gene mutations were found. And I started a nonprofit called the Rett Syndrome Research Foundation. And that was a really good time to get involved. Because the gene had been discovered and we were able to pull into the research people like Adrian, Rudolf Janisch, and others. We already knew a fair amount about the gene. We started off at RSRF. I was lucky enough that I was able to start the foundation with a handful of other parents. At the time, there already was a nonprofit, but they were family support focused and were not that interested in research, and suggested that I go start my own organization, which I did. And in the beginning, we didn't have a whole lot of money, and we were providing small grants to academic researchers in lots of different fields, because we really needed to kind of fertilize the field. And so we were funding biochemistry work and genetics and some clinical genetics and, lots of different areas to try to understand what the protein was doing, where was it found. We started to make animal models, a variety of different animal models to serve different experimental purposes. We're also growing our network of advisors. We were doing a yearly request for proposal. That's when we started out, we were maybe getting 10, 15 proposals. And with every passing year, that number grew. At the end, we were getting over 100 proposals for every RFP. Of course, that meant we needed a number of reviewers, key opinion leaders. So trying to grow that network. We were hosting an annual scientific meeting where we were bringing together the various labs. Again, it was all academic at the time, together for a couple of days to share data and to brainstorm and to really advise the Rett Syndrome Research Foundation on where we should be investing our funds and what could we do to stimulate more research. In 2007 came a landmark paper, again from Adrian, Kathy's husband, that showed that if you can restore levels of this protein, the symptoms in a mouse model dramatically go away. Everything that was measurable went back either to normal levels or almost normal levels. That was pretty unexpected. I don't know if I would say that it was unexpected for me. I was hoping, of course, that would be the outcome. Maybe it was just my need to have that be the outcome as a parent of a child that was severely disabled. But I think it really shocked the rest of the scientific world, clinical community, and of course the parent community as well. And I think it's become kind of the reversal, the experiment to do, and has been repeated now for lots of different disorders. not always with the same results, of course. I think Rett stands out as a disorder that's really remarkably reversible, at least in mouse models. We'll have to wait and see how that translates to people. I ended up leaving the Rett Syndrome Research Foundation for a variety of reasons and started the Rett Syndrome Research Trust in 2008. And the goal of the Rett Syndrome Research Trust was really to focus on genetic medicines. We have three main reasons for optimism. One, it's a single gene disorder, like your disorder is. The second is that it's not degenerative, right? The brain cells are all there. They're all in the right place. They're not communicating with each other properly. They're not functioning properly, but they're all there and they're not dying. And then we have this prospect of reversibility, which gave the community, a huge boost. And for me, it really changed everything. It made me really want to go after genetic medicines. Now, at the time, 2008, the technologies still weren't there. It was difficult to deliver to the brain. It still is difficult, but at least we have some technologies that allow us to deliver cargoes, genetic medicine cargoes to the brain. And so it just really made a lot of sense to go after genetic medicines as our focus. The field had matured. We knew a lot more about MECP2 and about Rett syndrome clinically as well. As an organization, we moved away from RFPs, requests for proposals, and moved towards, deciding which areas we wanted to be focused on. So, for example, we decided very early on that gene therapy was going to be a focus for us. Genome editing and RNA editing really hadn't been discovered yet. So there was really one way to boost levels of this protein, and that was through gene therapy. So we decided that that was really going to be a focus for us. We had other areas of focus, but the way we still operate is not to do an RFP, but to decide the areas that we want to focus on, figure out who's doing cutting edge work in each of these modalities, and then cultivate a relationship with these scientists and encourage them to submit proposals to us for funding. That's a big part of what I do day to day is really outreach, whether it's to academic labs or to industry, to really try to stay on top of who's doing what in the field and making contact and cultivating relationships with anyone that's doing work that's relevant for Rett syndrome. We started funding gene therapy work in 2010, and we funded a number of labs. And in 2013, there was another key paper from Gail Mandel, Brian Kaspar, and Adrian Bird, showing that in female mice who were lacking MECP2, that we could restore or reverse symptoms using gene therapy. So that was the first solid clue that gene therapy was something that could be relevant for patients. So at that point, I kind of took stock of the gene therapy landscape for Rett syndrome, who was doing what, and decided that it would be helpful to have a gene therapy consortium, because the situation that we found ourselves in, and I think this is common, is that you've got gene therapists who know a lot about how to deliver genes to the body. That is their area of focus, but they don't know anything about our particular disease. And then you've got people that know a lot about the disease. In our case, people like Adrian, for you, Kathy, and others, but they're not gene therapy experts. And so the thought here was, can we bring people together in a consortium with synergistic but different skill sets and get them to collaborate, provide funding, provide the infrastructure, provide opportunities to meet in person, provide opportunities for lab members to go to each other's labs and spend time and learn new experiments and techniques and share ideas. And we did that. We started a gene therapy consortium with 2 MECP2 experts and 2 gene therapists, and funded them from 2014 to 2017. And that was a very a great investment, because out of that gene therapy consortium came both of the gene therapy companies programs that are now in companies. We transition them from academia to companies Taysha Gene Therapies and Neurogene, and also an RNA editing program that started in Gail Mendel's lab and now is in a company as well. We're still focused on gene therapy, but we've also expanded now to other technologies, which I'll touch on in the next slide. As the science began to mature, we also started to have more engagement with biopharma. Typically, that engagement starts with me doing the outreach. Occasionally, we get contacted by companies as well, but it really takes quite a proactive approach of reaching out to companies that are in the gene therapy space or in the genome editing space, base editing, prime editing, these various ways to not supplement the gene by adding extra copies of the gene, but actually by correcting the mutations. The other thing that we did as the science matured and as we started raising more money was being able to hire staff that had industry experience. So when I started RSRT, it was pretty much me, but fortunately, we were able to hire people with industry experience, genetic medicine experience, clinical operations experience, and translational medicine. So we now have a team of 5 focused on the research. So these are the genetic medicine modalities that we're working on. Gene therapy, of course, is the furthest along. It's the technology that's been around the longest. There are 2 gene therapy trials, as I mentioned, 2 companies, actually multiple gene therapy trials, but sponsored by 2 companies. Still too early to tell, how well it's working. We're hoping that by the end of the year, both companies will be in a position to share some data. We're also working on base editing. So that's where we're able to address certain mutations that are caused by a single point mutation in a single nucleotide. Prime editing. So base editing was discovered by David Liu at the Broad Institute. He also discovered prime editing. I describe prime editing kind of as base editing on steroids. You're able to fix multiple mutations that are close to each other. Right now, that means really close to each other within, a couple of amino acids. But in time, this should be, as the technology matures and people get better at doing it, it should be able to address, entire, large sections of DNA, maybe entire exons. Because MECP2 is on the X chromosome, another approach of ours is to try to reactivate the silent MECP2 that's on the inactive X. And then we have RNA approaches as well, RNA editing and RNA transplicing. In transplicing, we're trying to splice out the 2 exons that have 97% of all of the mutations in MECP2 and splice in healthy exons. With the exception of MECP2 reactivation, these are all technologies, modalities that are applicable to your disorder as well. Then on the clinical research front, patient data and being able to assess symptoms in an objective fashion is also really important. We recently launched a Rett syndrome global registry. And so this is a registry where it's parent reported. So parents register, they register themselves, they consent on behalf of their child. And then they're asked a series of questions that we developed about symptoms and history, medical history, surgeries that they've had, also what doctors they see, therapies that they get, medications that they're on, what medications they think are helpful. So, a series of questions, and then they're prompted quarterly to go in and update. And then we have this digital natural history study, which doesn't cost anything. And it's something that I would encourage your group to look at. It's a company called Citizen, spelled CIITIZEN. As I mentioned before, they had been bought by Invitae, but then spun out and they're independent again. So it's a technology company. And the way it works is parents register on behalf of their child. They have to go through a registration process that proves they are who they are, and then they give consent for the company to solicit medical records from physicians. And you put in the doctors that your child sees, or the clinics that they attend, hospitals, whatever it may be. The more comprehensive the information you provide, the better it is. And then Citizen requests the medical records on your behalf. And then they have a machine learning program that allows them to extract information from, structured information from unstructured medical records that might be in PDF form, for example. And then the data gets shared back with the nonprofit anonymized in a spreadsheet form. And the thought process here is that if you're doing a traditional natural history study and you're doing a 10-year natural history study or a 15-year natural history study, that takes 10 years or it takes 15 years. And you're only getting a subset of people. You're getting the subset of people that are willing to participate, that can travel to a clinic, whose children are not too sick, or, it excludes families that can't afford to take the day off to go to a clinic, or can't travel to a clinic. So there's pros and cons of each approach. We have a traditional natural history study in Rett. This is meant to complement that and to add additional information, not to replace it. But this allows you in a very short period of time, as long as you can recruit patients, to get deep knowledge from the medical records. Because imagine how much information exists already in our kids' medical records. So Citizen is something that, someone could contact, and if anyone is interested, I can provide, I can give you the contact information of the person to reach out to. Our global registry we built with a company called Across Healthcare, and their registry program is called Matrix. This is a company that was started by someone who is very passionate about rare disease. He himself has a rare disease. And they kind of charge you on a sliding scale. So there are many patient advocacy groups that are for rare diseases that don't have a lot of money, who are able to build a registry almost for nothing, or very little money. And then organizations like ours that have a bit more money are able to subsidize because we're paying at at a higher level, and happy to do that, actually. So again, if someone is interested, I'm happy to make an introduction to Across Healthcare. Biosensors is an effort that we're undertaking to try to find objective ways to measure symptoms directly from the patient. What has happened in Rett syndrome is that the outcome measures that have been used in trials have historically been very subjective questionnaires that either parents have to respond to, or physicians. And because they're subjective, it makes them very placebo vulnerable. And we're looking to replace these questionnaires with objective outcome measures that are actually collecting physiologic data directly from patients. Things like sleep, seizures, breathing, movement. And so we're testing a number of devices, and we're in the process now. We're just launching a study that's going to be testing a device that we're very excited about, and that so far we've done a pilot study with, is something called the Emerald. It's a box that sits in a room, kind of like your Wi-Fi box, and collects data through radio waves. Our pilot study was focused on sleep and on breathing. And of course, they have to build algorithms, but it was really, really encouraging. And so now we're expanding that with expanding the number of patients. We're also going to be characterizing movement, and we will be comparing sleep and breathing using the Emerald while people are undergoing overnight sleep studies, which is kind of the gold standard And so we'll be able to answer the question, is what we think we're collecting with the Emerald actually what we are collecting? So we're excited about that study, and that's getting underway now at 3 clinics in the US. We also have a biomarker project, and the hope here is to be able to find something in blood that tracks with disease severity and would be able to give you an early signal of target engagement. So for example, in gene therapy, you might imagine that seeing improvements in with gene therapy is going to take some time, because it requires, a patient to learn skills. And a biomarker would be able to perhaps tell you early on whether you're on the right track. There's no guarantee that we're going to find one. We have some candidate biomarkers that look really interesting, and so we're chasing those. I think if we had a biomarker, it would make clinical trials easier. I think we'll see many more companies getting involved, but we'll see. There's no guarantee that we're going to find it. And I think biomarkers for neurological diseases overall has been a challenging field. And then finally, we have a biorepository. So these are patient cell lines, fibroblasts, and then a subset. We have 30 patient cell line fibroblasts, but then we also have 5 and soon to be 6, actually 7, the patient cell lines that we've made iPSCs, induced pluripotent stem cells. And we've had over 100 requests from companies and academic labs. The reason we made a biorepository ourselves is because although there were plenty of patient lines that had been developed in academic labs, we found that companies couldn't get a hold of them. So even if the academics themselves wanted to share them, it was difficult. Their institution wouldn't allow sharing in most cases with companies. And so we collected our own patient cell lines and funded the, making these various lines and iPSCs, and we're maintaining the repository at Coriell and handling the MTAs and things that are required. But it's been very, very helpful. So those are the clinical research efforts that are underway. I feel really strongly that organizations like ours are really important to get drug development and genetic medicine development moving in rare diseases. This kind of work just doesn't happen on its own. It's one thing if it's Alzheimer's or it's some other major disorder where there's a lot of incentive for companies to get involved. But for rare diseases, you really need somebody that's like driving these efforts forward, providing funding to labs and to getting companies engaged, and that laser focus on a cure. And really what we're doing is de-risking programs. So no company, Rett syndrome, and I suspect your disorder too, it's just not low-hanging fruits for companies. And there's tons of rare diseases. And, for companies to get involved, they ask a number of questions. How much is known about the protein? Are there animal models? Are there patient cell lines? What would we use for outcome measures? What could we recruit for a trial? Are there clinic sites? Is there a biomarker? And the more of those things that you can check off and say, yes, we have that. Yes, we have that. Yes, we've got patient registry. Yes, we have clinical data. Yes, there is a natural history study. The more likely it will be that companies, could be convinced to pick up a program. And so I think what we do is we de-risk these programs. We get them started in academic labs. We build the resources that are required, and then we help to try to make that transition from an academic lab to a company where drugs are typically developed. When I say drugs, I mean genetic medicines as well. We are technology agnostic at the end of the day, whether it's gene therapy or editing or something else that works. We want to see all of these efforts move in parallel forward as fast as possible. We think the more shots on goal we have, the better the likelihood of success. And we really act as matchmakers, between academics and industry. Cargoes and delivery, what I mean here is our genetic medicine cargoes, whether it's gene therapy or the machinery to do editing or RNA. And then for now, in terms of delivery, the gold standard is AAV9. That's what the 2 gene therapy companies are using, and that's what lots of academics and companies are using. In time, we hope that we'll be able to transition to a non-viral delivery method, because there are a number of advantages, immune responses being one of them. For now, you can only give AAV9 one time. Non-viral, and by non-viral, I mean things like LNPs, lipid nanoparticles, which is how the COVID vaccine is delivered, or exosomes, or some other non-viral delivery. They're cheaper to make, they're easier to make. There's no cargo space limitations as there is with AAV, and no or less immune issues. So there's a lot of advantages. Lots of people are trying to develop delivery technologies that get into the brain. We're talking to a number of companies and academic labs. We just started a delivery consortium to share ideas and to advise us on where we should be investing some funds. So I'm optimistic, but we're not there yet on the delivery. For now, we're depending on AAV9, and we'll have to see how well that works for our disease. And in terms of the clinical research efforts that I just described, the registry, the natural history study, I think those are efforts that are important to be advanced by a nonprofit who's going to make sure that everyone has access to them. If it's a particular biopharmaceutical company that develops, it ends up being proprietary and not shared. And so I think it's important for all of us to work on those and to make those resources available to anyone. So of course, that all sounds great, but it takes money, right, to fund all of this work. And I wish there was a magic bullet, but there isn't. All of our fundraising is really, grassroots, and the money that we have raised, every single penny of it can be traced back to families. That doesn't mean that all of our money comes from affected families themselves. Some of it does. The vast majority comes from their network, family, friends, colleagues, relatives, neighbors. And so we really depend on families to fundraise for us. And it's not easy, right? Because families already have a lot of challenges that they're dealing with. But unfortunately, that's the case. Anyway, I hope that was helpful, and I'm happy to answer questions.
Very interesting that you mentioned, and that actually somebody else, another researcher, had mentioned to me, is Citizen as a concept. I think that's something that we would be interested in trying to get set up, because one of the things that we've spoken about in the past is that we are really missing out by not having a functional registry now. And so I think Citizen is something that we're really going to investigate in the future. So I mean,
One thing I didn't mention,
I think you're
one thing I didn't mention with Citizen is so for families, once you provide this information and the medical records are solicited and you have them, you have a dashboard that you can access, and that you now have all of your child's medical records in one place, and you can access them and share them with whoever you want with a click of the button. So that's great for families. And then Citizen, if their business model is that if a company wants access to the data, they have to pay for it. And that's how they make money. So they don't charge the nonprofit, they don't charge the families, they'll charge the company that wants access. And then they share some of that revenue back with the families and with the nonprofit that recruited the patients. So, it's kind of an interesting business model, and I think a win-win for everyone.
Yeah, it does sound really good. And yeah, I don't think that there are any more questions in the chat at the moment for you, Monica, but if any come up, I can maybe pass them on to you, because we might get some after the event. But thank you so much for speaking. Really appreciate it. And I'm just going to share my screen quickly, just to close out the meeting. Okay. So I would just really like to thank everyone who came along to the conference. It was really nice to hear from all of you and to see, you were from all over the world. It was a really good turnout. And mostly I hope that everyone was able to get something from this. I also want to say thank you very much to all of the speakers who came along to talk and everyone who contributed to putting together the conference, because there's a lot of work behind the scenes from different people. And then finally, I just want to ask you, if possible, to please give us some feedback on the conference and what you thought of it, because it's really important for us to get feedback so we know what was good and what we maybe could improve, what we would need to change if we were doing other events in the future. So this is a QR code and I can post a link. In fact, I'll email the link out to all of the attendees afterwards, so you should get the link as an email. It should only take a few minutes to answer the questions, but you can answer as many or as few of the questions as you like. So if you just have one thing that you want to let us know, you can scroll straight to the end and say it. But any feedback you can give us is great. So once again, thank you so much for attending, and thank you to all of the speakers.